So I think we'll just start the session. So I'll be sharing my screen. Someone please let me know whether I'm audible and whether my slide is clearly visible or not. Just please let me know. So I think the sound should be all right here. So yeah, just please let me know in the chat whether it is clearly audible or not, and then we'll just go forward with the class. Okay. So I think we should be good here. So again, I welcome all of you to week 11's content for the NPTEL course Bioinformatics Algorithms and Applications, which is instructed by Professor Michael Bromia from IIT Madras. And I'm Sunanda Samant, a PhD scholar at IIT Indore, and I'll be taking the live tutorial session today. Okay. So in this week's lecture, as always, so whatever Professor Bromia covered, so for the tutorial session, what we will do is that we will try to solve some of the similar questions related to the concepts that Professor Bromia covered, which came in some of the earlier iteration of the course in the past couple of years. And then we'll try to go through that, and then maybe we will also try to intersperse some concepts and some talks like in between all of these numeric problems and the basically multiple choice problems. And also, so whatever Professor Bromia have covered, for example, uh, like he covered about model evaluation, model training, etc., some concepts related to those. And at the end of the lecture, I think there was also a tutorial for Autodoc, I think, for docking, right? So we'll try to uh, also like show a little demonstration of one other software. We'll try to do the docking with something called Autodoc Vina, which is not specifically by its native tools, but we'll try to do it via the integrated module inside UCSF Chimera, which we already have some exposure to as a visualization tool in some of our earlier tutorial sessions. Okay. So as a quick recap, basically what we did in some of the earlier classes is that we talked about, for example, QSAR, right, quantitative structure activity relationships, and how to basically how the whole workflow of the process basically works. For example, here we have dataset preparation, then molecular descriptor generation, then we divide that into training and test set. The training set is used for model training and learning, and then ultimately ultimately we have to do model validation after all of this. And basically, these steps can get us ready for a QSR model. So this QSR model is essentially a model where you can basically correlate the different features or the descriptors that you generate from your data from the small molecules, let's say, and then you kind of correlate that with activity, for example, binding affinity, something like that, right? So it is a very useful process that we covered in some of our earlier tutorials also. Again, we talked about the different kind of scoring functions which are used in various popular softwares, for example, Autodoc, ChemScore, Glide, etc. So there are, for example, force field based, empirical, and knowledge based potentials which are widely used for these different softwares. We saw some examples. We went over some details in a little, uh, basically covering the some of the algorithms for the or basically approach for the scoring functions, such as the empirical scoring. We talked about this, let's say, where the energy is kind of divided into these different forms or different terms. For example, energy for torsion, angle, hydrophobic interactions, ionic interactions, and so on. And then we talked about the force fields, which is basically the mathematical model to calculate the potential energy of systems atoms. And the force field is widely used, as we saw earlier, that not only the different scoring functions, but also in the case of, for example, classical mechanics. When we run molecular dynamic simulations through classical mechanics, there is a very important usage of force field, because it is essentially what allows us to calculate the different potential terms which are used to, ultimately to get uh, the coordinate of uh, the atoms for after a certain period of time, right? So in these calculations, the force fields are widely used. We learned about some examples, or not examples, just some classifications. For example, all atom, united atom, or force grained, whereas all atom was basically the force field describing each atom of the system in as its distinctive kind of unit. That was all atom, whereas coarse grained is kind of a more broader approach where not each atom is defined clearly, but just, for example, the entire residue or amino acid residue can be defined as a coarse grained bead. So this allows us to simulate a large system, but of course it will not have defined the very fine kind of information that a all atom model could give, right? So there are, of course, advantage and disadvantage to all of those. The more you use uh, the model that you use, if it is kind of trying to uh, sacrifice some of the balance, trying to sacrifice some of the accuracy that it has, then you can get better computational timing and so on. And these different algorithms, of course, have their own pros and cons. So yeah, so in the force field terms, we kind of quickly glossed over the different potential terms, for example, potential for bonds, angles, dihedrals, as well as improper van der Waals, electrostatics, and so on, which kind of, so these bonded and non-bonded potential terms kind of come together to form the ultimate, the potential equation for the force field based calculations, right? Then you also had knowledge based scoring functions, which we learned about that, for example, in PMF, uh, which uses basically this Boltzmann hypothesis, and basically calculates in terms of certain parameters the energy of a system, basically. So it can, for example, here we have given in terms of propensity, some energy difference is being calculated. Another approach is that when you have a large number of data points in a data set, you can divide the data points into basically particular sub ranges or beans, and then ultimately you can calculate the energy of the beans based on their populations. So the lowest energy bean will be the highest populated bean, and you can basically calculate the relative energy, which will give you an idea about the distribution of the data in terms of free energy, right? So that's also widely used in molecular dynamic trajectory analysis. Uh, can give us very useful insights in basically in which space basically our certain conformations are occupying, right? So in terms of different uh, collective variables, we can calculate these type of free energy calculations. We can perform this, right? Then we had just went over briefly about the genetic algorithms, where basically the uh, basically the initial population of chromosomes are randomly chosen. So here the chromosomes are particularly referring to a pose of a ligand inside a binding pocket. And then those different poses, so the high ranking poses are kind of combined together to produce the next generation or the next set of poses, which are referred to as the children here. And basically this process, after many iterations, will give you basically refined a binding pose inside the pocket, which could be assumed as the best kind of docked pose here, right? So these type of algorithms we just briefly went over. Uh,
So excuse me.
yeah, yeah,
So
please.
the previous slide, sir, that chromosome, does it refer to that actual chromosome, or is it something?
No, no, no. So this is basically referring to this genetic algorithm kind of as an analogous process, you can say, as our DNA replication and so on, right? So here it is not referring to the chromosomes as the literal chromosomes, but it is kind of drawing an analogy of the process from that, basically. So here, since the parent kind of poses, they are kind of combined to produce the next setup and the next generation poses. So they are best parts, kind of, you can say, for example, some bond rotation or some side chain orientation or something like that, some torsion angle. So these could be thought of as the features of the parent poses that are taken together to produce certain next generation poses or children poses. So because it is combining kind of the feature of the parents, some features of the parents, so that's why it is that's why they are kind of drawing the analogy of these chromosomes here. So that is also why it is called genetic algorithm, because it is kind of drawing an analogy from that. It is not literally trying to refer to the actual chromosomes, but just the process of taking features from certain poses and combining them to create a new pose, a more refined pose maybe. So just for that, they are doing the analogy.
Okay, sir.
Yeah, yeah. Uh, so yeah, we talked about these, and then we talked about we could probably represent the uh, experimental affinity values such as IC50, etc., in terms of their the different parameters. For example, we had simple as a simple example, we had hydrogen bond donor, hydrogen bond acceptor, and polar surface area, etc. We could just basically form equations, and then we could calculate the value of the variables, which would give us the uh, overall this, which could basically solve the problem in terms of that. Yeah. So, and we ultimately talked about just a little bit of computer drug design. For example, virtual screening and the molecular dynamic simulation, the first initial part of the entire drug discovery pipeline, right? And we talked about the current usage of artificial intelligence and machine learning, not only in the virtual screening and drug discovery processes, but the overall basically drug development pipeline, where each of the processes can be monitored or basically improved upon via the use of artificial intelligence, right? Yeah, so another kind of concepts that we went over, since we talked, we discussed about the IC50 values, right? So what are the IC50 values basically are, they are kind of the half maximal inhibitory concentration. So for example, if you look at a plot of something like this, where on the x-axis we have provided certain concentration of inhibitors. Here, for example, we have in the nanomolar range, and on the y-axis curve, if we have the remaining activity. So the point at which the y-axis curve, the y-axis value touches the 50%, right? So it has basically 50% remaining activity after applying a certain concentration of these inhibitors. So that will give us the IC50 value of the particular small molecular inhibitor or something that we are trying to add, right? We're trying to inhibit that with. So usually in the nanomolar range, in the low nanomolar range, the affinity, binding affinity is considered quite good, right? A lot of, I think, FDA approved drug and a lot of drugs that are kind of proceeding for the clinical trials and so on, or which are basically proposed as potentially valuable drug candidates, right? So those are having the activity at a very low range, basically, in terms of lower nanomolar range, right? So that is kind of the idea behind the IC50. Something like this we can also have, for example, at 0 minute, 30 minute, and then 30 minute. Basically under different concentrations you can get the activity results. So in this particular graph on the y-axis, it is written, I think, a little bit differently. So it is okay. So it was remaining activity. Here it is written as activity in terms of the percentage of control. So if you are comparing three different uh, environments or three different drugs, you can kind of use these type of plots. And then another type of representation is this percentage of inhibition. So it is the opposite here. So at how much concentration you are getting the percentage inhibition. So here, if the percentage inhibition kind of reaches 50 at certain point, then you can consider that as the IC50 values, right? So that was kind of what I wanted to introduce. And this one picture that I found from Wikipedia. So here it actually states that there is an error in the picture. So just kind of wanted to discuss that a little bit. Why is there an error in the picture? Can anybody mention? So if you look at the x-axis here, it has the log 10 base concentration, right? And as you increase, basically go forward with the log 10 base concentration, then you are getting this type of inhibition curve. But at this lower end, when you have a very low concentration of inhibitors basically present in the system in your target, right? It should not have the inhibition percentage or anything like that at 100, right? Because at no inhibitor, it should be mostly active, right? Or basically all the way active. And the more you add inhibitor, the more uh, the activity will decrease. So in this particular case, the y-axis should be kind of like activity or remaining activity. It should be represented in this way because at the lower end of the inhibitor concentration, it should not be highly active, basically, right? That was kind of the issue with this plot, which they have mentioned here, that the maximal inhibition would occur at the lowest part of the curve and minimum at the highest. So in this one would be the maximum inhibition, and this one will be the minimum inhibition essentially. So they have kind of made a mistake here, right? Okay. So in introducing or while talking about the different models that are used uh, to basically evaluate so different models and then the evaluation of these models and their performance. So during that conversation, Professor Promiha introduced this particular term, right, which is called ROC. So can anybody mention what does ROC stand for? So you can try to answer in the chat maybe. What does ROC stand for in terms of basically assessing the performance of a model, right? So since artificial intelligence and machine learning is on the high rise, right? So not only knowing about the different, let's say, algorithms that are widely used, not only is that valuable or important, but other than that, when you are applying the machine learning models in your certain work, in your particular project, and so on, how can you evaluate the performance of a model, right? So those kinds of things are also very important, and this particular term is basically kind of related to that, right? So you can try to answer in the chat, and we'll look at it in a moment. Okay, so I see one answer. B. Others can maybe also try. So here, essentially, when you talk about ROC in terms of assessing models and so on, so the ROC particularly stands for receiver operator characteristic. Okay, so receiver operator characteristic is what basically uh, ROC stands for. And if we kind of take a look at what it basically is, so it is kind of a plot where on the x-axis you have the false positive rate, and on the y-axis you have the true positive rate. Okay, and there is certain, let's say, a curve uh, that is maybe something like this. So that is kind of what an ROC curve typically looks like. And a lot of times you just calculate what the area under the curve will be, and that will give you an idea about how your particularly model is kind of evaluating or how your model is kind of performing, right? So we look at what these false positive rate and the true positive rates are, and the formulas that can be used to calculate these sort of parameters, right? Uh, but just from this idea, let's say if it is false positive rate versus true positive rate on the x and y axis, and where the area under the curve can give us information about the performance, right? Uh, so can you maybe try to answer this question just from that information? So the area under curve for a random ROC curve will be what? So for a random ROC curve, what could be the area under the curve? So maybe you can try to answer in the chat. You just look at it in a. Yeah, so I see one answer B in the chat. So yeah, let's just try to figure it out. So we know that ROC curve basically uh, basically it kind of coordinates the true positive rate as well as the false positive rate together, right? So what is the true positive rate, and how could we maybe possibly express this in terms of the data point present in our uh, basically overall calculation? So the data, by data point I means let's say how many true positives are being marked by the model, how many false positives are being marked by the model, how many true negatives are there, how many false negatives are there, and something like that, right? So what could basically true positive rate indicate? So if we express this in terms of the formula, so here it basically says, so it will be true positive divided by true positive plus false negative. Now, what this false negative basically means is that the system or the model is marking it as a negative. However, that evaluation is false. So it is actually a positive value. Let's say in a classification kind of problem. So you are trying to just figure out whether uh, just very, let's say in very simple term, whether this particular small molecule will bind to this particular uh, protein or not. So you only have a 0 or 1 answer, right? You only have a yes or no answer in that case. So kind of like a classification like problem if we treat it in that way. So there, let's say you have uh, false negative data, which will mean that the model that you have built, it is saying that it cannot bind to the protein, the molecule cannot bind to the protein, but it is kind of a false evaluation. So it is actually a positive marked data. It actually should be a positive data, basically, right? So that means this false negative is essentially a positive data. This true positive, that means that it was a positive kind of data, and the model also marked it as a positive data. So that is why they are called true positive, because actual positive data that are being marked by the model as being actually positive, right? So basically what the bottom part here, the denominator will indicate is that the total number of positive data that you have calculated on, right? Because you know the true positive means the positive data that are being marked correctly by the system, and this false negative will mean the positive data that are being marked as false. So total among all of them, basically, if you combine both of these together, so it will mean that how many positive data that was present in the calculation, right? So among them, how many true positives our system could get, that will be called the true positive rate. So for all of the positive data that are present, how many of them is marked correctly as being positive by the model. So that is the true positive rate. So similarly, can you maybe try to uh, answer what could be false positive rate? So in terms of these type of expressions, for example, true positive, false negative, true negative, false positive, these. So what could false positive rate indicate in this sense? So I think one answer, false positive divided by false positive plus true positive. So think of it in this way. True positive rate was among all of the positives, how many was flagged as true by the system. But in the case of false positive rate, uh, it should be among all the negatives, how many the system gets wrong. Basically, the system marks it as positive. So in that way, so if you write the expression in that way, so you would have false positive divided by false positive plus true negative, because this is trying to indicate among all of the negative values, how many negative values are being incorrectly flagged by the system or incorrectly marked by the system as a positive value, right? So here we have on the denominator, we have true negative. True negative means those are actually negative and the model also is evaluating them as negative. False positive means those are actually uh, negative, but the model basically incorrectly evaluates them as positive. So you have essentially total the overall negative on the denominator. So among all of the negatives that are being evaluated by the model, how many the model is being incorrectly marking as positive, essentially, right? So that is called the false positive rate. So essentially, uh, as we saw that the area under the curve for the ROC will basically have, let's see if we just drew one of the earlier slides what we did. So here you will have true positive rate, and then here we'll have false positive rate. It, let's say something like this, right? So when the ROC curve is random, what could be the relationship between true positive rate and false positive rate? If the evaluation by the model is kind of random, right? So that is what it is kind of trying to say. So in that case, what could be the relationship between true positive rate and false positive rate? So yeah, I see linear. Yeah, so basically linear will mean that in this particular case, your true positive rate will be equal to false positive rate, right? So if you kind of, let's say, think of it as an example. So if let's say you have equal number of positive and negative data, right? So let's say out of 100 total data that is being evaluated by your model, you have 50 data which are actually positive and then 50 data that are actually negative, right? So then your model, when it evaluates, so basically when your true positive rate equals to false positive rate, so what it will be something like this. So let's say out of those 50 positive data, the system is able to identify the 20, let's say, positive values as correctly, like true positive, right? Then out of the 50 negative data, again, the model is kind of trying to evaluate the 20 of them uh, incorrectly, basically. So as false positive. So in that sense, you will have your true positive rate equals to false positive rate. and that will basically indicate that it will be random, right? So because the system cannot correctly, or the model cannot correctly distinguish between uh, the positives and the negatives in that sense, right? So in kind of that sense, if you just draw the true positive rate and the uh, false positive rate, you will have a curve something like this, okay? And of course, in this case, if you notice that the area under the curve will basically be 0.5, right? Because the line is kind of dividing the entire square or rectangle into two parts, right? So that is kind of the concept here. So when your true positive rate and false positive rate, when that relationship is linear, or when your true positive rate equals false positive rate, so that is when we can say that the system cannot system is not performing well as it should be, which could give us a curve of something like this, okay? So in this case, the answer would have been 0.5, as mentioned in the chat also. So related to basically these types of terms, for example, true positive rate, false positive rate, and so on. So you also have heard of, while you know basically assessing models, we have heard of terms such as sensitivity and specificity, right? So what does sensitivity mean in this sense? Can anybody answer maybe in the chat? So sensitivity can also be expressed in terms of these values like true positive, false negative, and so on. So what could sensitivity indicate? So sensitivity basically is the same thing as true positive rate, okay? So basically what we saw earlier, true positive divided by true positive plus false negative. So of all the positive values that are there uh, uh, during the calculation, how many the system is correctly identifying as positives, right? True positives basically. So that is what is being referred to as sensitivity. Uh, false positive rate we saw is false positive divided by false positive plus true negative. And then there is another term called specificity. Specificity of a model, right? So what could specificity indicate in this sense? What is the specificity of a model? So the specificity basically can be also termed as true negative rate. And true negative rate is basically true negative divided by true negative plus false positive. So here, the false positive basically means that the system is identifying this data as positive. However, it is a false identification or false evaluation. It is actually a negative data, right? So that means that true negative plus false positive will indicate all of the negative kind of values that are present here, right, in the calculations. And among all of these negative value in total, how many the system is correctly, or the model is correctly identifying as true negatives, as negatives actually, right? So that is what will give us the true negative rate, or otherwise called specificity. So among all of the negative values, which can be obtained by just combining the true negative plus false positive, among all of them, how many the system is able to correctly identify as negatives. So that is the true negative rate of specificity. Uh, so just from this formula, can you perhaps tell what could be the relationship between false positive rate and specificity? So there seems to be a relationship between this false positive rate and specificity. What could that be? If we, for example, have a false positive rate reported for a model, can we evaluate its specificity? So maybe try to answer in the chat. We'll just look at it. So if you carefully notice, uh, both of them seem to be having the same denominator, right? This bottom part. So yeah, I see one answer in the chat. It is 1 minus true negative rate. So you can write it as this basically. So false positive rate will be equal to 1 minus specificity. Otherwise, 1 minus true negative rate. If you do it in that way, you will see that so 1 minus true negative rate will be 1 minus true negative divided by true negative plus false positive. And then on the upper hand, you will have basically true negative plus false positive minus true negative, right? So it will give you false positive divided by false positive plus true negative, which essentially is the false positive rate. So 1 minus specificity or 1 minus true negative rate will give you the false positive rate, right? Then there is another term while evaluating a model, which is called the accuracy of a model, right? So can you try to guess what it could be? What could be the accuracy of a model? So when we talk about the accuracy of a model, it is basically can be expressed via a formula like this. So we have correctly predicted values divided by the total number of positives and negatives that we have in our calculation, okay, or evaluation. So when you talk about the correctly predicted values, what it could be? So how can you maybe represent this maybe using some of these terms? What could be the correctly predicted values? So essentially on the upper side, the numerator side, we'll have true positive plus true negative, because remember, uh, when you talk about correctly predicted, that means that the negatives are also being correctly marked as negative, so that will be true negative, right? And when you talk about correctly predicted positive values, so that means the positives are being marked also as actually positive, right? So that will be a true positive. So the true values are the correctly predicted values. And on the denominator side, we will have the all of the positives and negatives combined together. So we'll have the true positive plus false negative, as well as the true negative plus false positive, right? So true positive plus false negative. Uh, this is basically giving us all of the positives that are in the calculations, and this true negative plus false positive. That means that this will give us all of the negatives that are present in the evaluation. And if you do this formula, so you'll get the accuracy of the model, right? So accuracy can also be used to assess a model, but a lot of times what people do is that people prefer uh, the ROC curve, the receiver operator characteristics, basically your sensitivity and the false positive rate, the curve between them. Because while a model can have high accuracy, let's say if you're trying to classify something between different among different classes, right? So a lot of time what happens is that the accuracy can be high. However, for a particular small particular class where the population is a lot low, so basically a minority population in that particular class, so it cannot always basically estimate uh, in a high basically performance level uh, the parameters for those particular classes. So that is one of the drawback sometimes that have been observed. And also, for example, one type of example that I came across. So basically, let's say in a banking system, right? So rather than having high accuracy regarding all of the transactions, it is more important to correctly identify fraudulent transactions. So in those types of way, the overall kind of performance, for example, something like accuracy, for that the accuracy may always not be suited, the most suited. So in a lot of cases, the sensitivity and the false positive rate, those are kind of uh, evaluated, as well as of course the accuracy and specificity and so on. So those type of parameters are also evaluated. So basically, not only the model accuracy, uh, but you have to kind of consider all of these parameters to exactly evaluate the model. So what its drawbacks are, and how it is performing on specific levels and so on, right? So that was kind of the overall, uh, basically, the prediction and the evaluation of certain models, right? We'll look at some of the uh, numeric examples related to these just a little later. So in another kind of set of concepts, Professor Gromiha basically went over something called awk programming, right? AWK awk programming. So in awk programming, so it is very helpful when you are trying to, for example, parse through a lot of data, right? So in a lot of cases, when you have large amount of data, so those data are generally text readable, right, or text editable. So they are in kind of text formats, but they have a very large file size and kind of a complicated arrangement inside that file, right? Different rows, different columns, and everything like that. So when we analyze molecular dynamics trajectories, for example, we also might get this type of output, because since it is having a large number of snapshots. So let's say 10,000 or 1 lakh snapshots we are getting from the trajectory, and let's say we are trying to calculate certain values uh, using those kind of snapshots, right? So we will have, let's say, 1 lakh total number of data points, or 1 lakh total number of rows, corresponding to which we have calculated, let's say, uh, different parameters, let's say, right? So that will be a large file. However, it is text editable, and so on. So what we can do is that we can use awk programming to quickly maybe try to get a certain information, or quickly parse through the files. In this kind of sense, it can become handy uh, to basically make the work process a little bit faster, right? So in awk programming, what character could be used to identify a field or a column? Can you answer that maybe in the chat? Just look at it in a moment. So yeah, I see answer. Yeah, basically, when you have to identify a field or a column, you can use the dollar sign in the case of awk programming, right? Now, can you answer maybe this question? So here it is asking that which of the following commands can be used to count the lines in a data file. So sometimes it can be very useful if you are, let's say, getting a certain output. You have to make sure that in the output file, the correct number of rows are present. Let's say the correct number of lines are present, right? So it can come in very handy whether, if you are trying to know whether your calculation have been completed or not, or it could have been terminated, right, in some unexpected way, right? So that is, in that sense, these kind of commands could be useful. So what could be the use of the awk programming while counting lines in a data file? Just look at it in a moment. I see an answer C in the chat. So we'll just look at it. Okay. So in this particular case, the actual answer should be uh, answer B. Okay. Now let's just try to maybe apply this one while trying to read through one of the data files that may arise, right? So just take a look at it. Yeah, I think it should be visible now. So let's say uh, we just uh, have this particular file, which is here. Let's just rename it maybe just for a little bit clarity. So it essentially what it is that it is one output from a molecular dynamics trajectory where first of all, if you open up the file, you will see there are three columns basically that are kind of mentioned here, right? So on the left hand side column, you will see this 1, 2, 3, 4, 5, 6, right up till all of the basically available data points that are present in terms of rows, right? So here, the left hand side column is basically saying that how many lines you have present in this particular file, inside this particular file. And as you can see here, we have a total number of 20,001 lines that are present, right? Uh, so the very first line here, we are seeing that some indicator is here. For example, here it is saying hashtag frame. So this would basically indicate how many frames that were present in our calculation. So in molecular dynamics calculations, what happens is that uh, whatever snapshots or frames that are present in a trajectory file, we just try to calculate certain values corresponding to those snapshots. So in this middle column here, so it is basically saying uh, which frames are we talking about. So it goes from 1 until 20,000. So it basically means that a total of 20,000 frames were present in this trajectory. And the third column here, we are seeing what we're basically seeing here is that certain value, which is the RMST value in angstrom associated with this particular snapshot. So here, let's say we are calculating just the protein RMST, which is indicative of the structural deviation of the protein from a reference structure, right? And based on whatever atoms you are considering in your RMST calculation, you might get different results. You might consider only C alpha atoms, you might consider only the backbone atoms, let's say C alpha, N, and C. And then you can also consider all of the heavy atoms. So excluding hydrogen, all of the other atoms you can also consider. So all of these will give you certainly slightly different results. But let's say whatever we have gotten here from a protein backbone RMST calculations. Now in this file, as you can see, a total of 20,001 lines are present, right? So let's say we wanted to just make sure that, okay, in our uh, basically calculation, the entire set of 20,000 uh, snapshots were considered, right? So what we could do, as we saw earlier, we could just run this awk command. Now we saw the awk command. So let me just write that down. So we could go basically one by one what this particular the commands that are given in the option what they do. So in our let's say first option that are given in the multiple choice, which are something like this, like so awk, then within quotation we have a curly brace, and then we have the print function or the print command, and then here we have this within this quotation mark we have /t, and then after that we have here $0, and after this we can just give the file name that we are wanting to get the information out of, right? So what it will basically do if we give this sort of command, So it should just, so we are just trying to, so I think I actually made a mistake while copying this data. So we just try to do the calculation for the original file only. So if we just give this command, what it will do is that it will show us all of the lines. So all of the lines that are present here were pasted kind of on the terminal. You can only see certain part of it because of course it's a huge number of lines there. But what this forward slash t essentially means that uh, these are being separated by a tab kind of type. So for clear visualization, a lot of times this forward t is used while getting the data in a lot of times you'll see, because it will provide kind of a more clearer visualization. Now, if we just paste, for example, the other command, what we can see is that, so let's just, instead of the print, we just see, we just print end here, and we can give, provide it like this, right? So this command was our answer B. And while running this command, you will see that the value 20,001 was pasted onto our terminal. So this is basically the number of lines uh, being counted on this data file. So that is essentially what it means. So that was this command, that was our answer kind of. And just to take a quick look at the other command. So for the print FNR and then $0 command. So what this will basically do is that it's kind of the same thing as the very first command that we used, but here we did not use the basically the tab. So that's why we are seeing that these two columns on the very first, they are kind of merged together. So, some sort of visualization problem, uh, which, to solve, we had to use the first command basically. And the last command is also very simple. So it basically, in this particular case, it will paste the entire file onto the terminal. But in that case, it had the command for only pasting the rows which does not have any blank values kind of type. So here we had, I think, awk nf greater than 1. So that means only greater than 1 values. So basically only non-blank values were pasted onto the terminal in that sense. So in that way, you can see for certain rows whether some data file are missing or that some data points are missing or not. So maybe we can just switch back to the presentation here. And yeah, let us try the next set of questions. So we just learned about, we talked about, the sensitivity, specificity, and so on while assessing certain models, right? Now, in this particular question, it is kind of a numerical question related to that only. So here we have the experimental set of values, right? So the experimental values are sort of indicated by, in a classification problem. So it is indicated by N or B. And in the question, it is given that B is positive, right? So that means the N will be negative. And on the other hand, we also have the predicted calculations for those data points basically. So in this question, you are being asked that what could be the sensitivity of the model. Now, remember that sensitivity was equal to the true positive rate, right? So true positive rate indicates true positive divided by true positive plus false negative. So essentially what you have to calculate is that uh, by looking at this experimental and predicted data points, we have to first identify which of these belong to the true positive category, and then also which of these belong to the false negative category. Okay. So true positive means we are only talking about positives that are correctly being marked by the model as a positive value, while false negative means that it is marked as a negative by the model. However, it is actually a positive value. Okay. So could you try to maybe do the calculation quickly and answer in the chat? We'll just look at it in a moment. So we should first try to identify among all of these actual experimental values and the prediction values, how many can be categorized as true positive values and how many can be categorized as false positive predictions, right? So we just take a look at it. So basically, true positive will mean that among all of the actual positive values, so actual will in this sense refer to the experimental values here, and of course since B is being referred as positive values, so we can say that in the experimental values whenever B was present, those will be positive, right? Now among all of the B that we can see in the experimental value, how many were correctly identified as B in the predicted set of values? So here we can see 4 B's which were in experimental as well as in predicted values, corresponding to, so they are basically the same, right? So B versus B, that's correct prediction for positive values. And then we also had false negative rate, which is basically, okay, so we also had one BB here and one BB here. So in total, we can say that the true positive values were 6, right? For the false negative rate, it would have indicated among all of the positive values, how many were categorized by the model as a negative value, incorrect, right? So that means we have to take a look at firstly which p value was there in the experimental set of data, corresponding to which the model gave us uh, a value of n, right? So what could that be? We are seeing one set of data here, which was in experimental set identified as a p. However, in the predicted set, we can see it written as n, right? So that is one false negative prediction. Another one we can see, I think, here. So the same B versus N prediction is being made here. So I think in the total, we can see 2 false negatives, right? So basically, this would indicate, I think I also see one answer. I think I see one answer C in the chat. So in this case, it would basically come down to, I think, 6 versus 8, right? So 6 versus 8 is 3 by 4, so it is 0.75. So in this case, the answer would have actually been 0.75, which means that the sensitivity would have been this one, option B. Okay, so that's how we can calculate these sort of things. Now, could you try to calculate the specificity maybe? So we just learned about the specificity uh, evaluation, right? Calculation. So its formula, if you remember, it was something like this. So true negative divided by true negative plus false positive. So true negative means the model is marking it as a negative and it is actually a negative value, right? So it is correctly flagging this data point as a negative value. Whereas false positive was indicating that it is incorrectly being marked as a positive value, whereas it will actually be a negative. So from this formula, what can we get as the value of the specificity here? first we have to identify the true negative and false positive values, right? And then we can just put that together to get the answer. We try to calculate it and share in the chat. So if we just take a look at the answer here, it should give you around 0.625, 0.63, right? So firstly, what will true negative indicate? So that means these are actually negative. So in the experimental set of data, we'll see it marked as N, right? And they are correctly being identified as N in the prediction by the model, right? So we can see this category of data is kind of being the first two data we can see here. These two, I think, also the same, and then this. So in total, we are seeing a total of 1, 2, 3, 4, 5. So 5 data point which was actually negative and correctly predicted by the model as negative. And then conversely, if we take a look at the false positive uh, prediction, so that means that in the experimental set of data, those will be negative. However, on the predicted set of results, those would have been incorrectly flagged or incorrectly marked as positive, right? So we can see these three prediction which can be categorized into false positives. So in total, we get 5 true negatives and 3 false positives. So we can get the specificity of 5 divided by 8, which will give us an answer of 0.625. Okay. So if the questions like this come into your assignment, so you can use these simple formulas to calculate the sensitivity and specificity. Okay. Yeah. So for our original question about the sensitivity, it was 0.75 or 75% answer. So the next question is kind of asking that, so if you have a correlation between two sets A and B, and you have that as -1, right? So basically they are perfectly anti-correlated kind of. So which one of these expressions can correctly define that relationship? So you seem to have A equals B plus 0.5, then equals minus B plus 1, equals 1 by B equals 1 by B squared. So here it is saying the correlation between two sets is -1. So kind of like perfectly opposite correlation or anti-correlated, right? So you can think of it in this way: if the value of uh, one sort of set increases, then the value of the other one drops. In that sort of sense, you can also think of it. But here, since basically it is giving us -1, so I think that would be a kind of a not correct way to judge it. So in this particular case, the answer actually is A equals minus B plus 1. Okay. So not the value drops, but the value kind of becomes negative in the opposite direction, kind of, right? So in that sort of sense, you can uh, get the answer of B. So equals minus B plus 1. Okay. So in that sort of way, it can basically get the relation. It can give the relationship of a linear anti-correlation, that kind of thing. So this one will be a reciprocal or inverse square relationships, the C and D answers. But you are trying to get a linear correlation, so that in that case you will get an equation like this. Okay. So it is kind of becoming the value in the opposite direction if you're thinking about a vector or in a direction kind of way. Uh, so that is sort of this question is implying. Okay. So along all of the concepts regarding different models and then evaluation and so on. So Professor Gromiha also very briefly, quickly, went over uh, the advanced sort of machine learning models, right? And introduced something like deep neural networks and so on, right? So here the question is kind of asking uh, simple concept regarding that. So we know that the neural networks have different layers, right? So how many layers does a neural network have to have for it to be classified in that category? So basically, in a neural network, what is the minimum number of layers that you can have that is required? Kind of. So I think also a question that was asked in some of the earlier course assignments. So in one of the earlier iteration of the course. After these questions, we'll just quickly try to see how a talking procedure mainly works. So Professor Promiha, I think, went over in an extensive kind of way about how Autodoc works, I think. So we'll just try to see a very simpler implementation of that via the UCSF Chimera, the molecular visualization tool that you also saw in some of our earlier classes also, right? So we'll try to implement it through that program. But yeah, in this particular question, what would be the number of layers in a neural network? So it is actually three, right? So for a neural network, of course, you need to have the input layer and the output layer also. But you also have at least one or multiple hidden layers. So in this case, the question was kind of asking for you to include this hidden layer, at least minimum one hidden layer in the neural network, and count it as the total number of layers, basically included in that as well. So we have essentially a minimum number of three layers in a neural network. Okay, so you can take a look at these particular uh, videos that I have linked here, which could give you a better idea regarding these concepts. Okay. Now, since we just calculated the problem regarding the specificity and sensitivity, right, earlier also. So here we have a very similar type of question, but presented in terms of a matrix, which is often called a confusion matrix. So here we have the actual values on the upper hand side. On the left hand side, we can see it is the predicted values, right? So for the columns of the actual values, we have again positive values and negative values. For the rows, which indicate the predicted kind of side, which also have the positive and negative values. And that junction points kind of will give you whether the prediction was true positive, true negative, false positive, or false negative, right? By considering the junction point of these two, uh, the actual versus predicted here, which are divided into these different columns, right? Positive and negative columns. So in this case, what could the specificity value be? So we know that specificity is calculated by true negative divided by true negative plus false positive. So can we quickly try to calculate from this matrix here? So these sort of numeric questions might arise in your exams or also in your weekly assignments, because Professor Pamela kind of went over some of these type of questions, right? So just you need to remember these formulas. So basically what specificity is trying to trying to say, what sensitivity is trying to say, and how can we represent those in terms of the true, negative, false positive, and so on, right? So these formulas, just remember these, just try to understand that, and then you can apply very easily in the assignment problems. So in this particular case, again, if we just go over it, so the answer would have been 0.8 here. Now, true, negative, this category of values, these are basically originally the negative values, which is this one, right? This negative value here, and also correctly predicted as being the negative value. So these 20 values that have been given in this cell, so these will come under the true negative category. Whereas for the false positive category, we will see that the values that were actually negative but incorrectly predicted by the model as positive. So we'll have 5 values that were false positive. So if you do the calculation, you will get 20 by 25. So it will give you 0.8, right? Specificity. So in the uh, answer, basically in one of the earlier iteration of the courses, so I think in the past year or the year before that, so they kind of gave the answer in a range here, so 0.75 to 0.85. However, you can get the calculation using this formula as well. So here, uh, as you can see, it is kind of falling just in the exact middle of the range, right? So anyway, uh, again, a very similar question, but here we have a sort of a more descriptive uh, question that we need to answer. So it is now asking about a protein DNA complex where the protein has 100 amino acid residues, right? And among these 100 amino acid residues, we are given that 15 residues are forming a binding site. Now you're trying to use a prediction program of, where you are trying to predict the binding site using the program. And the prediction program identifies 20 binding site residues. So it was actually having 15 residues, but the prediction site has identified, a prediction program has identified 20 residues are being part of the binding site. And only 9 of these 20 residues that was predicted by the program, they are matching with the experimental data. So experimental data is what we have to consider as the ultimate data, or the ultimate truth here, right? So the experimental binding site residues are considered as positives, essentially. So from that, what would we say about the true positives and false positives? So how many true positives were in this prediction and how many false positives were in this prediction? So maybe try to answer it in the chat and let's see. So basically, again, you would have to identify what were the positive uh, values in the experimental, what were the positive values that were identified by the prediction model. So those will be the true positives, right? Because it was in the experimental set of data also positive and it was predicted as positive by the model. And then for false positive, you will have basically what is being incorrectly identified as positive, incorrectly being identified as positive by the model. So essentially in this case, the answer would have been 9 and 11. Now, how do we get here? Uh, so basically, you will see that among the 20 binding site residues that were predicted by the model, 9 were matching with the experimental data. So these 20 binding site residues, that means they are being evaluated as a positive, right, by the model, or predicted as positive by the model. And among them, only 9 are matching with the experimental data. That means these 9 were also positive in the experimental data. So these 9 are essentially positive in the experimental data and correctly identified as a positive by the prediction model. So here we will have the true positive number as 9. So that means of the 20 binding site residues, other than 9, the 11 other binding site residues that were predicted by the model, that was incorrect prediction, right? So those were being marked as positive by the prediction model. However, those were not binding site residues. So those were not present in the experimental data. So that means these 11 are actually negative values, but being identified as the model by positive values. So that means these will be false positives. So in this particular case, we had basically true positive value of 9 and then false positive predictions, total 11, right? Uh, so 9 and 11. So what could the sensitivity of the method be? We know that it is true positive divided by true positive plus false negative. Now here we are not talking about the false positives which was asked in the question, but we are trying to calculate the false negative here. So false negative means the model is correct, model is predicting the data as negative. However, they were actually in the experimental set of data, so they were actually positive, right? So you can get that uh, if you see that the 9 residues that the model predicted were matching with the experimental data. So that means there are 6 other residues that are present in the experimental data that were not being identified were not being correctly predicted by the model, because initially we had been given that 15 residues were forming the binding site which are provided by the experimental data. So if the model identified 9 among these 15 correctly, so that means 6 other residue was remaining there, which the model could not identify as part of the binding region, by part of the binding site. Right? So that means those 6 values were the false negative values. 15 minus 9, 6. So those were the false negative values. And if you do the calculation here, so that means the true positive was 9. Uh, so you divide that by true positive plus false negative, which is 9 plus 6. So 15, and then you will get the value of 0.6 sensitivity. Okay, so it is 60% sensitive in that sense. Okay, so in the next set of question, again we had one about the awk command line. So here it is asking that if the data file has a value of more than 57, so it will only consider those lines where it has a value of more than 57 on the 5th field. Okay, so these fields are kind of indicative of columns that we saw in our files also, right? And those are being indicated by this dollar signs. So which option could it be? You can just probably guess it from the general representation of the comment itself. So if we have to get the lines which has a value of more than 57 on the 5th column or the 5th field, which could it be? So it will basically be here in this case the first one, because as you can see here, so within this quotation mark, what is basically written? So it is taking the $5 or the 5th field or the 5th column, and it is kind of applying a condition that it is greater than 57, right? And then it is printing that out on the terminal, right? So that means this one will indicate wherever the $5 or the 5th field has a value of 57, so those lines will be given as the output. So these kind of this question, I think I also discussed in one of the earlier slides. So here it is asking that for classification problems, which is used as a best measure of evaluating the performance. For classification problem, which could be the best measure of evaluating the performance. I think it was also discussed by Professor Gomiha in the lectures also. So could you maybe try to answer in the chat? So among all of these, the proper answer here will be the sensitivity and specificity. Okay. So here we also have the accuracy as an option, but as we talked about, one of the earlier slides is that a lot of time accuracy might not uh, correctly evaluate the model basically. So wherever you have a classification problem and where you need to basically divide the data points into different classes, uh, what happens is that classes with minority kind of populations are not always being correctly adjusted for something like that. So in that case, the accuracy a lot of times does not give the best evaluation, but the sensitivity and specificity they do. So in those cases also, these sort of things are these sort of parameters are taken into account. Okay, so we'll just uh, take a look at one of the demonstration. Professor Kumiha went over Autodoc, I think, and then we'll just try to see a very simpler implementation of that through the use of the Chimera software. So I hope the screen is visible, right? So here, what I have on my local system uh, is that we are trying to, let's say, dock a small molecule inside a protein, right? So let's say this is our protein. So whatever protein it might be, right? So let's say this is our target protein, and we have to dock the small molecule inside this binding cavity. So you can see that already one ligand is kind of present inside this cavity. If we just take a look at the surface visualization, maybe it will be a little bit more clear. So let me just change it up a little bit. And let's just show the surface here. Yeah. So we can see clearly that the ligand is sort of uh, present inside this cavity that is being formed, right? So this is basically the binding site or the binding region for this small molecule. And we earlier talked about one sort of way you can identify which exact residues are part of the binding region, right? So we discussed about the 5 angstrom sort of approach. Let's just hide the surface quickly first. So what we also could do is that we could just select the ligand in this sort of visualization software. And for Chimera, there is a very handy uh, command here or very handy selection process here. So you can select a zone through a UCSF Chimera command line you can use, or the GUI you can also use. So basically what we can do is that we can select the ligand molecule, and then we can basically select a 5 angstrom zone around this ligand molecule. So whichever residues, amino acid residue is being present within a range of 5 angstrom from the ligand, it will be selected here, right? And the same can be seen if you open up the sequence. So we'll see uh, the residues that are in the vicinity, basically within 5 angstrom, right? And you can just hover over with your cursor in the sequence list, and you can see the exact residue and its numbering being given. For example, here we have ASP 52, glutamic acid 53, arginine 54, and so on, right? Uh, Now that is one way to identify the binding site. But when you're trying to, uh, let's say, dock a small molecule, so of course during docking, during this process of docking, you need to clearly indicate or mention the binding site where you are trying to dock, basically, right? So in one process, you have to basically make a grid that kind of defines the site within which you are trying to dock the particular small molecule or ligand here. And in a lot of softwares, you can mention these in different ways. For example, you can provide exactly which residues are part of the binding site, or so you can make a grid around that region. Or let's say in some cases, if there is already one experimental structure where a co-crystallized ligand is present, so you can take that position of the ligand, which is already present in another co-crystallized structure, as a reference, and you can sort of build a grid around that, right? So in this particular case, we will just try to do exactly that. We can just clear the selection first, maybe. And which molecule we are trying to talk, we'll just try to load it up to just show you. So firstly, just uh, yeah, firstly select the protein, and then basically hide all of the side chains that are visible, so that it will make the visualization a bit more clearer. Uh, and the molecule that we are trying to dock will be, just wait a minute. Uh, so what I was trying to do is, let me just open it up in a different way. Okay. So from that co-crystallized ligand, what I have actually done is that I have just eliminated the ligand that was already present. So that gives us the protein, which will be used as our target in the docking protocol, right? Uh, and then I will just open up two different ligands here. So one, you will see this ligand.pdb, which I have opened up here. So it was the co-crystallized ligand that was originally present in a PDB file. Okay. Now we will use this ligand as a reference point, and we'll just try to build a grid around this in this docking protocol. And then the other ligand molecule that I want to dock should be present here. I think it is just overlapping the proteins somewhere. Hmm. Just wait a minute. So here, this ligand.pdb that I had opened up earlier, so it was part of a co-crystallized ligand. We'll use this as a reference position to build our grid around this. And as you can see, there is another small molecule that I loaded up here uh, on this particular visualization terminal, you can say. So this particular small molecule, which is being referred to as this hashtag one molecule here, or the ligand 2.pdb, which I had opened up earlier. So this is the molecule that we are trying to dock onto this position, right? So we have loaded up this molecule. So essentially what we have to do when you go through a docking procedure, so Professor Gomiha, what he mentioned also is that there are several steps, right? Firstly, you know that in experimental crystal structures, the hydrogen atoms are missing usually, right? So you have to firstly add those hydrogen atoms uh, wherever applicable. Then we have to add proper charges, right? You have to calculate the charge for the ligand also. And then after that, you have to basically prepare a file for the grid that will basically describe the grid. So grid is basically a point of reference for where your small molecule is going to be docked into, right? So for that reason, we have basically taken this co-crystallized ligand uh, here also. We have loaded it up here. So we'll just use that as a reference point. So for each of these individual steps, what you can do is that you can go to this tools bar of this program, uh, and during basically under the structure editing part, you will see that a lot of options are present, right? So you can, for example, add hydrogens here. So you can add polar hydrogens here maybe, and after adding the hydrogens, if you again go to the structure editing part, you will see the add charge here. So for that, you can calculate the charge in the form of AM1 BCC here, right? So this is one protocol for charge calculation and so on. So you can do these basically individually and try to prepare the files. There is doc prep option also available, I think, under the surface and binding analysis. But you will notice that in the tools bar under the surface/binding analysis, there is one very handy option which is called Autodoc FINA. So Autodoc FINA is a separate program, of course. You have to download it and install it in your system, maybe, right? Yeah, so if you take a look here, you will see that this is the site for the Autotalk Vina. So it is from the Scripps Research Institute. So you will see the vina.scripps.edu basically website where you can get the particular software, right? And now here you will see that the different options are available, of course, depending on whatever your operating system is. So for me, of course, since my operating system is Linux, where I'm trying to run this command, so I had downloaded this particular basically package, right? So this one, the very uh, top particular option, right? Uh, and what we basically got from that is this compressed file here. So this was what was downloaded, and then when I extracted this, so there are this bin folder along with a license kind of file. Inside this bin folder, we had the executable. So here, the main one that we will be using is this Vina one. Okay. Now for the particular program to work through the usage of Chimera uh, software, you have to give the path of the executable. So for me, in my system, the executable is lying inside this path. So I can use a Linux command called print working directory to get the uh, particular folder path. You can use similar commands in a Windows terminal also. You can use those type of commands, right, to get the path. But anyway, what we basically can do is that uh, inside this particular tab for the Autotalk Fina, so we can firstly describe the location of the output file here. So let's say I just want to be inside the parent folder where I was originally keeping the files, and let's say we'll just give it a name as output. Uh, Notice that it has an extension of PDBQT, so that means that not only the coordinate but the charge information is also being kept here. And now it is asking us to give the receptor file and as well as the ligand file. Now for our, of course, receptor, it was named as receptor.pdb. For our ligand file, we have to mention whichever one of these we want to dock. So this was the co-crystallized ligand in the magenta or pink color. The blue one was the one that we actually wanted to dock, right? So I think this is ligand 2. Uh, in the option. So we can give that option, and then after that you will see that one option kind of is given here, which is called receptor search volume options, right? So under this we will get this option resize search volume using button 2. This is why we kind of needed a reference point to calculate our grid. So if you already know whichever area this binding pocket is kind of in, so you can just do it. Uh, you can just make a grid in that region. But here, since we have a nice point of reference as the previous ligand, we can just use this. So what you can do is that firstly, you can just press control and select the entire ligand that we want to use as a reference. And from your mouse, you can just press the middle scroll button, and you will notice a very handy cube being generated around the ligand reference that you want to take, right? Now, this cube is basically representing the dimension of the grid where you want your molecule to be docked. And also very handily, what we can do is we can shrink or extend the sides, all of the sides of this cube using again by pressing your the middle scroll button of your mouse. Let's say I just want to make it kind of uniform from all sides, and so that it can cover a decently wide area, so that if we have a ligand of a different size, that can also be accommodated inside this particular region. So let's say we just cover this side, we just extend a little bit of that. So upon extending all of the sides, we can get a grid something like this, right? You can just move around the grid, again similarly using your uh, mouse as well. Now, let's say after we have clearly defined the grid in the 3D space, and when we have made sure that all of the sides are kind of covering the small molecule, okay. So now do not adjust the grid's position anymore. But what you can do is you can again select the co-crystallized ligand, the previous one, which we only wanted to take as a reference point for the grid generation. We can just delete it from our uh, terminal here. And so that leaves us with the protein, the grid, as well as this ligand that we want to actually dock here, right? Uh, you can go back to the Autodock Vina tab that was earlier open. You will see that the center of the grid and the size of the axis are also given here. So these are automatically generated and being modified as we made changes to the cube grid. Uh, you could have, of course, put this particular information right from the get-go. If you had information regarding the center and the size of the grid, basically you could have put that information right from the beginning. But anyway, after defining that, so here we had a receptor as receptor.pdb, and then ligand as ligand2.pdb. So this one, which was opened up earlier, and then a lot of things are given basically in the different kind of options. So here, of course, since it is kind of a workflow type of thing, so you do not need to exactly do the individual steps by yourself, so separately, but it has a nice integrated options for all of those different steps. For example, here we have add hydrogens, then merge charges, remove non-polar hydrogens, then merge charges and remove ion pairs, and etc. So on and so forth. Here we also have given ignore waters as true, because we do not want to, in this particular case, we do not want to, uh, we do not want there to be any water molecule that can obstruct the binding pocket in a way. And for the ligand options, of course, you have given the charge options here. For the advanced options, you have been given how many binding poses or modes you want to ultimately get in your output file, the exhaustiveness, the maximum energy difference, etc. So all of these options for the receptor and ligand options, it is very nice that it is given a kind of under a simple umbrella. So you can use these, you can basically tick these boxes. Um, and then for the executable location, you have to give the path for which your executable file is located at. So in my computer, of course, as I showed, it is in this path. So it is residing in this path. We have the executable, and the executable name is Vina, right? So I have kind of given that path as a whole here. And if you just press OK, you will see some text on the bottom here that it wrote some of the files that are being generated during this docking protocol. And then it is currently running. So it should take maybe a couple of minutes. It's only a single molecule, so it's not that time consuming. So let's see what the result brings us. Okay. So it was very quick. Of course, depends on your CPU's performance as well. But in general, kind of in modern PCs and modern laptops, if you're trying to just dock single molecule or let's say 10, 20 molecules, it should not take that much time, right? You should get the result relatively quickly. Now, in this case, we had just gone with the default options that are present in the preparation kind of steps, right? However, when you try to use like Autodoc Vina or MGL tools or the Autodoc their tab, right, in that parent kind of program, so in that you will have a lot more exhaustive kind of options that you need to give. So you need to take each of these steps, preparatory steps, etc., seriously into consideration. But yeah, anyway. So yeah, you need to consider all of the steps kind of critically. But ultimately here in this particular simple process, we got these results here. Okay. Uh, Now you will see that it has generated a bunch of different conformations for the ligand, and you have a score corresponding to the docked pose, right? So the docked score or the docking score, we know the more negative it is, the better indicator of a binding affinity. Basically, the better binding affinity it is being indicated. And of course, after that, you have the RMST calculation. So you have two different types of RMST are kind of being mentioned here. But anyway, in any case, the best score, you will see that the RMST corresponding to that pose is given as 0. So that means that it is taking this particular pose as a reference point to calculate the RMST of all of the other poses. So that's why for the other poses, you have certain RMST values. But for the best score, you have a RMST of 0. So because that pose was taken as a reference, so of course, comparing with itself, it will have a deviation of 0, right? But anyway, so if you just press on this different kind of options, so first of all, you have, let's say 7.4 here. So if I just make it this way, so let's increase it a little bit to visualize the binding region. So if you just click on this very top score, you will see that it has a conformation like this, of course. Now, of course, all of the other scores does not mean that because those are also negative. So it's not necessarily that those scores will bring you the same sort of conformational orientation as the best scoring ones, because if you just click on the minus 7.1 option, you will see that it has a different orientation, right? So these two rings were in different position. In this position earlier, and the side chain was also here. In the next pose, you will see that it is kind of flipped in a way, right? But anyway, in that pose also, you will see a minus 7.1 score for the docking. The other poses, if we just quickly go through them, you will notice that the pose is being different, right? However, they seem to have a decent docking score. So one thing that you can gather from this is that the docking protocol is not the most accurate evaluation of a binding, right? So that's why what we do in in any in silico drug discovery protocol or something like that. So if you have gotten hits from the docking protocol, the virtual screening protocol, to get the proper orientation or whether there is any dynamic change in the orientation during in a certain environment or not. So for analyzing these kind of things, you have to pass this docked molecule to a molecular dynamics simulations protocol. So you have to calculate from a molecular dynamics trajectory through certain algorithms, for example, MMPBSA, MMGBSA, or in case if you want very high accuracy, but very highly computational, cost heavy, right? Highly resource intensive. So you can also go for the absolute binding energy calculations, for example, free energy perturbation or thermodynamic integration, such things. But in general, approaches like MMPBSA and MMGBSA are quite standard, so you can use those. But anyway, you have to calculate from a trajectory and take an average of that and see what kind of deviation it is giving you also. So in that way, you will get kind of a better representation of the binding in terms of the binding affinity than the docking, right? So for an in silico process, you have to kind of procedures. That is kind of now the standard. You have to follow up your docking with uh, a simulation sort of study. And after that, if you get hits from the simulation, then you can proceed for any further studies, for example, wet lab experiments and such. Yeah, so you are getting, for example, these different poses, but if you are trying to proceed forward with another study, for example, molecular dynamics study, so it is better that you take probably the minus 7.4 docking score, minus 7.4 kcal per mole. That scoring pose here. Uh, Another way to get a proper pose will be that if it was already having, let's say, co-crystallized structure, and then your novel molecules are kind of also similar in structure with that co-crystallized ligand. So you can compare their poses. So the co-crystallized pose versus the novel ligand pose, and see whether you are finding similar sort of orientation or not. So that is another way. But in general, what a lot of people do is they just select the minus 7.4 kcal per mole, or the lowest sort of binding free energy, the best sort of affinity score here, and they proceed forward with other studies like molecular dynamics simulations, because anyway through that study they will get the dynamic nature very easily, like if the ligand is changing conformation, if it is orientating in a different way. So those sort of information and their corresponding binding free energy calculations will be obtained by those MD simulation studies, right? Anyway, you can get that, and you can save these PDB file via this file and save PDB option. So you basically have to first look at, okay, which was your um, model or post that you wanted to save. So in this case, since this is the first one, I believe it should be the very first post, right? Um, and you can save that along with the receptor file here. So let's say we are trying to save these two. You can very easily save that. You can save in a single file, maybe. Yeah, you can just give a file name here. Let's say some random file name here. You can save in a single file, and it will be saved basically very easily. So then you will get the docked protein ligand kind of complex, and you can proceed forward with another studies if you wish, because we now have the exact coordinates of the ligand inside the binding pocket, right? So that's one simple approach that I wanted to mention. Of course, while actually performing experiments, you need to properly follow a standardized protocol and such things. uh, you need to refer to different literatures, that what kind of parameters they have taken for their different calculations. So based on that, you need to decide which sort of parameters you want to use in your, let's say, ligand preparation, protein preparation, and so on. You need to understand what could be the drawbacks of selecting these type of parameters as compared to this other value, something like that. So you have to basically consider all of that while you are actually trying to perform the experiment. This was just a basic kind of representation of that. Okay. So yeah, I think we'll just wrap up the class today. So that was kind of all of the sets of questions that I had. So yeah, so thank you everyone for joining the class today. And again, in the next week's tutorial session, we'll cover the next set of lectures. So please try to attend the other TA sessions as well if you have the time available. And then also try to go over Professor Komiha's lectures very carefully, because the questions in the assignments and the exams that we will get will be kind of relating to those concepts that he covers in his lectures, right? Okay. So again, thank you everyone. Uh, and then we'll meet in the next class. Okay. Thank you.